All set now. We are live and we've started recording and we can let people in.
Hello and a very warm welcome to all of you who are tuning in from all over the world. We are live on the platform of Biologically Speaking. We're almost at the very end of our conference, Her Story of Science, celebrating women in research. We're celebrating the phenomenal contributions of Indian women in research, and we are very delighted to have you all joining us on the eve of International Women's Day. We're going to wait for just a minute or two to get everyone in from the waiting room to the Zoom meeting. Meanwhile, I'd like to take this opportunity to introduce our platform, Biologically Speaking. We are a group of doctoral and postdoctoral trainees from all over the world with a common passion for science outreach. We are a platform organizing regular webinar series showcasing the frontiers in global biological research. And not only this conference, I sincerely hope that you all will stay with us beyond for all of our future endeavors. All right, so I think we are all set and we have everyone who were in the waiting room currently in the Zoom meeting. So without further ado, I'd like to introduce today's keynote speaker, Dr. Gayati Hassan from the National Center for Biological Sciences, Bengaluru. Dr. Hassan is a professor and served distinguished fellow in NCBS. She has had an illustrious scientific career spanning over 3 decades, and I'll try to briefly summarize the vast body of impactful scientific work of hers. Dr. Hassan graduated from the University of Cambridge with a PhD for her work on ribosomal RNA genes of Trypanosoma brucei. In 1983, Dr. Hassan joined the Tata Institute of Fundamental Research in Mumbai as a visiting fellow and research associate in the molecular biology unit. She then joined as a founding member of the National Center for Biological Sciences in Bengaluru in 1989, where she serves till date. At TIFR, Dr. Hassan completed the genetic and molecular mapping of an olfactory mutant in Drosophila. During her time at Brandeis University, she initiated work on Drosophila intracellular calcium signaling by cloning and characterizing 2 Drosophila genes for intracellular calcium channels. At TIFR Bangalore, her laboratory pioneered the methods of reverse genetics to generate the first mutants in the Drosophila gene for the inositol 1,4,5 triphosphate receptor, and continues to work on intracellular calcium signaling in Drosophila growth and neuronal functions. Her lab aims to understand the role of intracellular calcium signaling and its implications in human disease. Professor Hassan has received numerous awards for the recognition of her groundbreaking work in the field of neurobiology. She has been a recipient of INLAC's Foundation Scholarship. Amongst others, she has also received a fellowship from the Wellcome Trust and a Rockefeller Biotechnology Career Fellowship. In 2006, she was elected a fellow of the Indian Academy of Sciences, Bangalore, and in 2007, as a member of the Asia Pacific Molecular Biology Network. She is also a fellow of India National Science Academy, and as of 2019 has been elected as the Vice President of INSA. She is also on the editorial board of journals such as the Journal of Neurogenetics, Invertebrate Neuroscience, Current Science, and an associate editor with Frontiers in Molecular Neuroscience. Dr. Hassan, we are truly delighted to have you on our platform, and the screen is all yours.
Well, thank you. Thank you very much. I hope everybody can hear me.
Yes, I think they can.
Okay.
Yeah.
Wonderful, wonderful. Good evening or good morning or good afternoon for all the people wherever you are in this world. I am absolutely delighted to be on this meeting. It's a really a pleasure and as well as honor to be here to actually speak in a meeting organized by younger people I don't think I've ever had this opportunity before, and I'm really, really very happy to be here to be doing this. Okay, so I will now move on to talking about our work. As you can see, and as was described in the introduction, for which I'm very grateful, by the way, thank you. Our lab works on neuronal calcium signaling. And we have worked on this for many years now. And I would like to now take you through what our findings have been. I will not go through too much of the past work, but essentially try and explain to you what a summary of what our work has sort of achieved over the last 30 years. So when we think of calcium in neurons, I think most of us think of the uh, image that you're seeing here, which is that you have calcium entry, you have action potentials coming down the neuron, voltage gated calcium channels are activated, and they let in calcium. This calcium then acts on synaptic vesicles, and you have neurotransmitter release, which acts on the synapse, and then you have continuation of the neuronal signal in the postsynaptic neuron. What I would like to talk to you about is another mode of calcium entry, which is this one. This is called intracellular calcium signaling, and it's somewhat different from what I described to you before earlier. In this, you have all cells, all metazoan cells, contain intracellular calcium stores, and also a molecule called the IP3 receptor or the inositol 1,4,5 trisphosphate receptor. Now, this acts as a calcium channel and as a ligand-gated calcium channel. It responds to extracellular signals. GPCRs bind a ligand, and this ligand then activates the GPCR. You get activation of G proteins, and as a result, you get cleavage of the molecule PIP2 on the membrane into IP3 and DAG. Now, IP3 binds to the IP3 receptor, and calcium is released from here. Following this process, once calcium is released from the ER store, you get depletion of calcium in the ER store. And as a result, there's this molecule called STIM, which is present on the ER membrane. It senses the drop on calcium and then activates a channel called ORI, which is present on the plasma membrane. Now, this mechanism is called store-operated calcium entry. I will come back to all of this later, but I just wanted to give you an idea of what we mean by intracellular calcium signaling. So as you can see, this process is rather different from what is described here. The question that we've been trying to understand for many years now, and for which I hope we can give some answers, is that if this process is present in neurons, then what does it do? What does this mode of calcium signaling actually do inside neurons? Is it the same as what happens here, or is it different? Does it do different things inside neurons? This is something that has been of enormous interest to us, and this is what I would like to talk to you about in the subsequent part of the talk. Okay, so what is the significance of intracellular calcium signaling in the human, in humans, or why is it even of interest? Apart from answering a question in basic science, loss of intracellular calcium signaling in Purkinje neurons leads to neurodegeneration, which you can see here in the human brain. So this is the region of the brain called the cerebellum. It's in the hindbrain, and this region, as a loss of intracellular calcium signaling, degenerates. The Purkinje neurons are integral component of this region, and they integrate multiple classes of information for motor control. So degeneration of Purkinje neurons leads to a neurological disorder. And this disorder is shown here. I would like you to just look at this slide. It's called spinocerebellar ataxia. So SCAs, which is a short form for spinocerebellar ataxia, are mostly caused by inherited mutations in a number of genes, including the type 1 IP3 receptor, which I described to you a little earlier. Type 1 IP3 receptor, loss of type 1 IP3 receptor, just one copy loss, causes SCA15, SCA29, and a more complex disorder called Gillespie syndrome. Change in intracellular calcium signaling has also been observed in other more common and sporadic neurological disorders like Parkinson and Alzheimer. So I think what I'm trying to bring home to you is that not only is the understanding of IP3 signaling in neurons important in the context of understanding a basic question, but it's also quite relevant in the context of disease. Sorry, I'll move on. Okay, so here are some of the mutations that are for the IP3 receptor 1 gene, which have actually been discovered in Indian patients. And what I wanted to bring home from this slide is that firstly, human mutations are seen in a number of SCAs in India, and they span across the IP3 receptor protein. So this is a molecule, this is the IP3 receptor shown here. It has a ligand binding, a modulatory, and a calcium channel. And as you can see, these mutations are present everywhere else. Secondly, there are regions in the human IP3 receptor that are highly similar to the IP3 receptor in the fruit fly, Drosophila melanogaster. So these regions are shown here. The ones in red are closer in sequence. And interestingly, amino acids that mutate in the human IP3 receptor 1 and which cause disease are actually conserved in the Drosophila IP3 receptor. So telling us then that using the fly IP3 receptor could actually be something that we could use to study basic functions of the IP3 receptor, and that would actually help us understand what is happening in humans. So we have been studying IP3 receptor function in fly neurons for the past 30 years. Due to its size, the fruit fly is amenable to molecular, cellular, and genetic manipulations. I hope to convince you that model organism studies can provide new leads on how to treat complex neurological disorders like SCAs. And given here, as you can see, is the difference between what a fruit fly brain consists of and a human brain consists of. So one could think that perhaps, what is the point of studying the fruit fly brain? It's so small. But as I said, I hope to convince you that this actually does give us some valuable insights. So here's an image of a fruit fly brain. What is shown here is the sort of the anterior part of the brain. And one of the things that one can do in Drosophila, and which one can do very beautifully, is to target specific neurons. So here, these are dopaminergic neurons, which have been targeted with the fluorescent protein, GFP, green fluorescent protein. And you can see how beautifully they light up and they can be seen. So there are many other neurons in this brain, but using genetic tools, we can actually just mark very, very few neurons indeed. And this is a big advantage to the kind of studies that we were interested in doing to try and understand IP3 receptor function. Okay, so what happens if you mutate the IP3 receptor in Drosophila? So here is a wild type fly on the left, which is can fly normally. And what you can see on the right is an IP3 receptor mutant. And given the same stimulus, which is an air puff, you can see that the IP3 receptor mutant is unable to fly, even though it otherwise looks perfectly normal. Now, remember, this is an IP3 receptor mutant, which is a partial mutant. So it is not a complete loss of function. Complete loss of the IP3 receptor actually leads to lethality. But these are mutants which reduce function but don't completely lose it. And in these mutants, you can see that the flies are unable to fly. So our interest then was to try and understand what is causing this. So here I'd like to just introduce to you the flight, or what we would think the flight would be doing, how flight would be affected. So flight generally starts by receiving sensory stimuli such as visual, mechanical, and olfactory. These reach the central brain, which is present here, and then they are integrated, and this information is then transferred down to the ventral ganglion, which has something called the central pattern generator. The central pattern generator neurons then coordinate muscles, flight muscles for wing movement, and this initiates flight and appropriate flight maneuvers. So when we found that the IP3 receptor actually prevented flight, it was possible that it was affecting the muscles, it could have been affecting the neurons in this region. It could have been affecting neurons in this region, or it could have been affecting wing development. I mean, anything was possible. So we really needed to figure out exactly what happens when the IP3 receptor mutants are affected, are not showing flight. To understand this, we use the genetic tools that I talked about, and essentially then looked at neurons and other tissues to try and figure out where IP3 receptor was important. Now, I won't go into the full details of that work. It's all published and it's all old work, but basically we found that it was a set of central neurons that were required that required the IP3 receptor. And as I said, this was using methods that allow us to sort of knock out the gene and do things with it with specific neurons. I would now like to just introduce to you again what the IP3 receptor does. So this is what happens when you have neurotransmitters. Neurotransmitters will bind the G protein coupled receptor, activate phospholipase C, and as I said, generate IP3, which will then bind to the IP3 receptor and release calcium. And this leads on to store-operated calcium entry, as I described earlier. Now, to really understand what happens with which cells are important for flight and how the IP3 receptor actually affects function in these flights, in these cells, Anamika in the lab generated a version of the IP3 receptor, which is called dominant negative. It's a genetic tool, and it stops IP3 mediated calcium release in specific neurons. This IP3 receptor is nothing but a mutated version of the Drosophila IP3 receptor, in which 3 glutamine residues essential for binding IP3, so they are in the ligand binding domain, are mutated as shown. The functional IP3 receptor is a tetramer, which is shown here. And it consists of 4 IP3 receptor subunits. All 4 monomers need to bind calcium for calcium release to happen. When a monomer of the dominant negative of the dominant negative version is present within the IP3 receptor, the tetramer cannot release calcium. This was demonstrated actually earlier by a mammalian group run by David Yule, and they had shown that all 4 monomers are absolutely essential. So we decided to use this method to actually try and understand whether if we express such a dominant negative in specific cells, then can we actually affect flight, and then use this to actually identify the precise cells that require IP3 receptor function. So we tested flight after dominant negative expression in a number of neuronal subsets and found that a cluster of neurons, which are shown here, the PPL1 neurons, these are dopaminergic neurons, they require IP3 receptor function. Dominant negative expression, IP3 receptor dominant negative expression in these neurons affected flight, whereas if we expressed it in any of the other neurons that are shown around here, for example, these ones, their flight was completely normal. So we could focus on this particular cluster. Anamika then dissected the PPL cluster even more. This was again, as I said, due to the wonderful genetic tools that are available for Drosophila, which allow us to do this kind of dissection and actually go down to single neuron level. So she dissected it and found that there were 2 neurons, which are shown here, which were actually essential for flight. So I'll take you through this data a little carefully. So here what Anamika has done is, in the first case, which is this lot of neurons here, I won't go into the name, but this is actually most of the PPL1 neurons which are present in this cluster. When you express IP3 receptor dominant negative, you find that flight is significantly reduced. So this is the time, flight duration. Here is what the control looks like, and here is what happens when you express the dominant negative in it. When she did it in these specific 2 neurons, you can see that flight is significantly reduced, almost similar to what you see here. But if she expresses it in any of the other neurons that are present in this cluster, which is shown here, then you can see that flight is normal and pretty much similar to the controls. So this was a wonderful, actually, piece of detective work done by Anamika, and it helped us to actually pinpoint neurons which required IP3 receptor function in a very specific way for flight. Now, these 2 PPL1 neurons, which are shown here, project to a particular part of the brain, This is the region here. This is shown here, which is called the mushroom body. And in that, a particular region, which is called the gamma 1 peduncle region. So the question then was that how and why does the IP3 receptor in these two neurons, dopaminergic neurons, which project to this part of the central brain, how and why does this actually affect flight? And this is what I'd like to talk to you about next. So for the first thing that we needed to do in this context was to understand how IP3 signaling in these neurons is activated. And as I have shown you before, this requires activation of a G protein coupled receptor. So what from some of our previous work, we knew that the muscarinic acetylcholine receptor, which responds to acetylcholine, is required for flight. Anamika tested whether these cells respond to muscarinic acetyl, whether they contain the muscarinic acetylcholine receptor, and if they respond to acetylcholine, or rather a version of acetylcholine, which is called carbachol, which only stimulates this receptor. And she found that indeed they do get activated by it. So then she looked at a very nice way by using a new tool. She then looked to see whether this addition of carbachol, which is shown here, what it does to these neurons. I will just go back for a minute and show you the region that she is. So this is the region that she is now imaging, which is the region where these neurons send their synaptic endings. So this is shown here. This is in the mushroom body. And Anamika, now what she did was she imaged this region for dopamine release. So this is a fluorescent sensor that is called GrabDA, and it increases its fluorescence when dopamine is released. And as you can see, in the wild type neurons, when you add carbachol, there is an increase in fluorescence in this region. What Anamika could show was that when you put the IP3 receptor dominant negative in those neurons, in those two neurons, then this level is considerably decreased. So you do not get significant dopamine release. And this finally led us to understand that one of the things that the IP3 receptor does is that as a result of signaling, it reduces dopamine release in the mushroom body, and this leads to loss of flight. Now, I think one of the questions that we really got interested in at this point was that what is the significance of these shorter flight bouts? What do they mean to the fly in a sense? Does it actually have a meaning? And for this, in this context, Anamika did some very interesting experiments. So what she did was she had looked up in literature and found that these two neurons had actually been discovered earlier by another group. And that group had shown that if you stop activity in these neurons, then if you take a hungry fly and put it in with food, so shown here is the food that we are talking about. This is a pellet, this is a bit of fermented yeast. Then a hungry fly will walk around and find this food in a few seconds, and then it will stay there and keep eating it. We wondered whether this kind of test, whether these neurons, given that they were already required for food searching, we wondered whether expression of the IP3 receptor dominant negative in these neurons would affect this kind of food searching behavior. And so Anamika tested this flies. This one is expressing IP3 receptor dominant negative in these neurons. And what you can see is that this fly, even though it's hungry, it's been starved for the same amount of time, it just walks around. It never actually finds the food and doesn't seem to reach there for a very, very long time. And here is the quantification for this kind of work. You can see that with the IP3 receptor dominant negative, flies take much, much longer to find food, as unlike the ones that are normal and which have the normal version of the gene. So this was a very interesting finding, and it sort of helped us put in context what this means. And this is a nice summary slide which Anamika made to try and get this whole message across, which tells you that when flies are flying along, if their dopamine signaling in the brain is affected, then it affects their motivation. And basically, even if they're hungry, they don't go and they don't bother to reach the food. And so we think that this is a way by which flies integrate both, the sense of hunger and sensory stimuli, and then use that to actually continue flying. As you can imagine, I mean, flight is a very energy intensive mechanism, and flies will not want to fly just for the sake of flying. So this is something that is very interesting, and I'll sort of try and move on from this work now to try and take you back to what I had originally talked about, which is try and explain to you how IP3 signaling affects in the context of neurodegeneration. Okay, so in that context now, one of the things that we thought about, which would be interesting, is that perhaps this what I discussed so far was acute intracellular calcium signaling, which modulates flight. But to understand what happens in the long term as a result of changes with IP3 receptor signaling, we postulated that perhaps one of the things that might be happening is that there might be an altered gene expression, and this might be the reason why neurons undergo neurodegeneration with time in the case of altered intracellular calcium signaling. So for these studies, we have targeted all three components of IP3 signaling, which is the IP3 receptor, ORAI, and STIM. And for the sake of time, I will show you our results with STIM primarily, which are the most sort of conclusive and hold together in across organisms. So this is the key data from one of the first RNA-seq studies that we did in the lab with STIM knockdown in pupil neurons. One of the genes that we identified in this RNA-seq is RAL, which is shown here. It's downregulated when you downregulate STIM in Drosophila neurons, in pupil neurons. And interestingly, what we found was that overexpression of RAL in STIM knockdown, in ORAI knockdown, these are all neuronal knockdowns, in IP3 receptor knockdown, or in an IP3 receptor mutant, if you overexpress RAL, you can see that flight is rescued to some extent, and this is shown in all these cases. So telling us that indeed RAL, the fact that it was downregulated actually would have an effect on the flight and on neurons itself. So what does RAL do? RAL is a small G protein, and what it does, what Shlesha, who did this work, showed was that RAL is actually a small G protein, and it's known from work in sort of other systems that RAL acts as a small molecule that binds the exocyst complex and is involved in vesicle release. Now, this work was primarily known in non-excitable cells. And what Shlesha showed was that RAL actually helps in vesicle release and synaptic vesicle release as well. And in the case of impaired RAL, when not enough RAL is present, this part is affected. So you get reduced levels of synaptic vesicle release. So in a sense, what we found was that even gene expression studies essentially led up to more or less the same kind of phenotype that we were seeing in the acute phenotype that I had shown you earlier, which was directly affecting dopamine release. What Shlesha has shown then is that RAL gene expression through intracellular calcium signaling also affects dopamine release and leads to loss of flight. So this is shown here. So with this work, which I described primarily about Drosophila, I'd like to now switch and talk a little bit now about what we have done in the mammalian context. So essentially, as I said, gene expression seems to be something that we thought might be important. It does, in the context of flies, it definitely affects the expression of genes that are required for function of neurons. And what we wondered was whether does intracellular calcium signaling regulate gene expression in mammalian neurons? And do changes in gene expression impact function over time? So for this work, I will switch now to talking to you about the mouse brain. So this is work that has been done by Dhanya in the lab. And what I'm showing you here are Purkinje neurons in red in a section of the adult mouse cerebellum. And this is what the cerebellum looks like. And this is a section, and this is a high magnification image of these Purkinje neurons. So what Dhanya did was she generated a mouse in the lab where she could knock out STIM specifically in these neurons. So again, using genetic tools, she used something which allowed her to knock out STIM specifically in Purkinje neurons. She then very carefully and painstakingly showed that in these mice, you can actually lose calcium signals if you activate these Purkinje neurons with an agonist for the IP3 receptor, and which also affects STIM signaling. So this here is your STIM wild type, which you can see the calcium signaling in signals in these Purkinje neurons, and you can see here in the knockout, these signals are completely lost. So having validated her knockout, Dhanya then went ahead and did a gene expression study. And for this, she microdissected the Purkinje neurons shown here, made lysates with them, and then did a RNA-seq, just like I described earlier for STIM in Drosophila. She found a number of genes which are upregulated and downregulated. I won't, for the sake of time, get into this in detail, but I'll just show you a summary of the kind of genes that she found which were affected. There were genes that affected neuron projection development. There were genes that affected post-synapse organization. And so essentially telling us again that IP3 or intracellular calcium signaling in this case is definitely involved in over time in affecting the way that genes are expressed, which then affect neuronal function, because all of these mechanisms that you see here would essentially affect neuronal function in some way or the other. Okay, so she validated these genes next, and this is shown here. Gene expression changes in Purkinje neurons with loss of STIM1. So some of the genes that are shown here are for neuron projection development, calcium binding, and ion channel. She did this by qPCR. And what I now want to show you is that we were able to actually take these genes and using a genetic tool, which I will now describe to you in a minute, but I want to first show you the result, we were able to revert these changes. So all these changes happen in 1-year-old mice. Okay, and what you can see here are these down regulation is all in 1-year-old mice. Now these 1-year-old, and they are actually not seen in younger mice. So if you take mice at 1 week, then the changes are very, very minor. So this is something that happens over time with knockout of STIM1 in Purkinje neurons. Now here's a method by which we are able in 1-year mice to actually bring back these genes to normal expression. So how did we do this? This is again something that had been going on in the lab. I won't go into the details of it, but what Dhanya did was to include a gene for this molecule called septin-7. What we had shown in the lab earlier was that septin-7, which is forms a part of a cytoskeletal filament, that if you remove septin-7, then you get more calcium entry into the cell. So breakdown of septin-7 reduces these filaments, and these septin molecules are known to regulate calcium entry. And you can see here that as a result, it brings in more calcium into the cell. So, and this is shown actually, I'd just like to show you some images here. So you can see here, this is an IP3 receptor RNAi control. Here's an IP3 receptor RNAi where you can see calcium release and then calcium entry, which is shown here, the greater flow, which is considerably reduced as a result of knockdown of IP3 receptor. Now, in this condition, if you reduce the level of septin-7, you can see that this is restored. And these are the graphs showing you the same thing. So essentially, we knew that septin-7 is able to restore back calcium into neurons. When you introduce septin-7 in the background of STIM1 knockout, we were able to show that we could regulate back genes to a certain set of genes were back to normal. And then what Dhanya did was to check if these mice actually could function better as well. So one of the things that STIM1 does is, you can see here, that mice are not able to hold on to this rotor rod because they lose their ability to coordinate motor function. And what you can see, I'll show this to you again, this video. What you can see is these two mice here, which contains the STIM1 knockout, but also have a heterozygosity for the septin-7 gene. You can see that they are able to perform much better than the STIM1 knockout. And I think the next slide shows you. Okay, so this is basically what we are looking at now. What we found was that STIM1 knockout, normal levels of septin-7, you have reduced intracellular calcium signaling, you have reduced gene expression, which is what I had shown you earlier, and you have reduced motor learning, which is shown here. Now, when you bring down septin-7, your intracellular calcium signaling goes up, which I showed you in the previous slide. Your gene expression goes up, which I showed you in the earlier slide, and you also have better motor learning. And this motor learning is what we quantified here. So you can see this level of motor learning goes up. So as you can see, this is really exciting in terms of looking at disease, because what we are saying then is that septin 7 is a molecule which changes intracellular calcium signaling, and as a result you can actually bring back some level of genes that probably are involved in neuron projection development, synapse organization, and calcium homeostasis. And the mechanism, as you can see, is through this. So one of the things we are still trying to figure out at this point is what is this transcription factor that drives this, the upregulation of these, or the regulation of these genes. And this is something that is still an open question, and is something that we are interested in addressing. Okay, so I would then like to just go back to the original question that I had raised, which is, in the context of human neurodegeneration, a dysregulated intracellular calcium signaling may be a consequence of altered age-dependent gene expression. And the genes that we have identified in this process could possibly serve as therapeutic targets for neurodegeneration. Now, I'd also like to mention at this point that even though spinocerebellar ataxias are relatively rare diseases, they are rare genetic diseases, but to this date, there is actually no therapy available for them. So anything that one can find in this context would be of interest to the several patients who suffer from these debilitating degenerative diseases. And as I said, these diseases have also been of late, there are several of them now found in India as well over time with sequencing. So there is a possibility that this work could actually lead to something that would help in a therapeutic fashion. Okay, so I'd like to finish with that. I hope I've been able to convince you of the use of model organisms for studying human disease. I'd like to now introduce the people who did this work. So Shlesha is the one who did the earliest RNA-seq work that I talked about. The IP3 receptor dominant work that I mentioned was done by Anamika, who's present here. Siddharth and Shlesha together did that RNA-seq work, which I talked to you about. And Bipon and Triambak were together involved in the septin 7 work. Finally, here is Dhanya. She is the person who did all the mouse work that I described to you single-handedly. And other people in the lab, Ranjita, who's here, and Pragya, and there are some others who are actually not in this image. They are working now on working with human stem cell neurons to try and develop actually the model of spinocerebellar ataxia in a dish, so that we can actually start doing some of this work directly in human neurons. I'll stop at this point, and I would be very happy to take questions. I'll stop sharing. No, not stop, sorry. I'll stop the thing and show. Okay.
Thank you, Professor Hassan. We really enjoyed your talk. I loved how you started. It all started from like watching mutations, and then starting from the flight being affected to going to the dopaminergic neurons, and then how connecting it back to the food-seeking behavior in flies. And not only that, you also looked into mice, which is great, like looking into two different systems. So we have a couple of questions, and with your permission, I'll start asking them.
Certainly, I'd be happy
Yes.
to answer them. Yeah.
Thank you. So first question is from Antara Banerjee. She is asking, why were the IP3 receptors in the dopaminergic neurons specifically targeted for mutagenesis?
So we had figured out that this flight phenotype that we see, it can be rescued by putting the wild type IP3 receptor back in dopaminergic neurons. So we knew that dopaminergic neurons were important in this context. And so that's the sort of the past history of this work. And that is what sort of allowed us to, continue to begin sort of looking at this, specifically the dopaminergic neurons.
Thank you. Akshata is asking, are there other compensatory mechanisms for calcium signaling in these neurons, Also, can there be a rescue conducted with AAV approaches?
With, sorry, what kind of approaches?
Double AV, the vector.
Oh, that's the with the yeah. No, so certainly, yeah. No, there are so okay. I'll answer the first part. There are almost certainly compensatory mechanisms that operate. For one thing, I should mention, and I didn't go into the details of it, that there is more than one IP3 receptor gene in humans and in mammals. Unlike Drosophila, where there's only a single gene, there is more than one IP3 receptor gene. There's more than one STIM gene, and there's more than one ORI gene. So for one thing that could definitely happen is that when you have downregulation of a certain set of genes, one particular isoform, other isoforms can be upregulated. So there can be compensatory mechanisms. And indeed, probably one could use this kind of neuronal injections with the AAV to try and improve function. We come from a genetics lab, so we have essentially used mostly methods of genetics. So in fact, the Septin-7 knockout that I described to you was also done using a genetic method. But certainly, I mean, viral injections can also be used, and they could probably function. I mean, we are not a mouse lab, so I guess that was one of the reasons why we chose to do work with genetics rather than with injections. But it's possible.
Thank you.
Yeah.
Thank you. Vani Pandey, who is also part of our platform, she is asking, is there any other behavioral changes apart from food-seeking behavior in flies?
It's very likely that there are. And it just means that one needs to look at other behaviors, which we have not done. I mean, I think, our focus has been on trying to understand the flight and how flight is affected. And once we found that, we wanted to know the context of it. So one of the things we looked at was food-seeking behavior. But it's very possible that in fact, these neurons have been found to affect other kinds of behaviors as well. And on the top of my head, I can't remember what they are. But so it's and those behaviors could be tested by expressing IP3 receptor dominant negative in them. So, but all I can say is we don't know at this point, but it's very likely that yes, there are other things that are affected.
Thank you. Bhakti Pathak is asking, is food-seeking behavior also rescued by expression of RAL in IP3 dominant negative mutants?
We haven't tried that. Yeah, we haven't tried that. Yeah. It's possible that it might be, but we don't know. I should mention though that the RAL phenotype actually seems to be affecting more strongly at the pupal stages. It seems to work there. So we don't know how important it is in adults, in the adult stages. The IP3 receptor dominant negative we expressed in the adults and showed what happens there. Whereas RAL expression has been done in pupil neurons. So it may or may not work, but it's something that would certainly be of interest to look at. Yeah, but we don't know. Yeah.
Okay. We also have a question from Professor Nahid Ali. She was the keynote speaker for the other day. Thank you, thank you, Professor Ali, for turning up today's session. She's asking why IP3 mutation only affected a particular neuron and not all as a whole.
So I think it's not that it doesn't affect other neurons. It's just that it's a bit of it's an interesting question actually. So we think that the IP3 mediated calcium signaling is involved in modulation of neuronal function, and flight is one of those functions which is very, it requires a lot of energy, and so it's modulated very, very carefully in Drosophila. Because a fruit fly will not fly just for the sake of flying, because it takes up too much energy. So it will use it, conserve it very carefully. So I think the reason we pick it up we pick up flight as a phenotype is because of the fact that it is so carefully neuromodulated, and that is why IP3 mutations show this phenotype quite easily. Remember what I said earlier, that IP3 receptor mutants that we are working with are not complete mutants. This reduces the level of calcium release. They don't completely block it. And in fact, if you completely block IP3 receptor function by using an allele which we call a null allele, then you don't actually get adult flies. They die as larvae. They die as early larvae. So it's a question of gradation. And possibly other functions that require IP3 receptor are not affected because that the level of calcium release that is there in those neurons is not so critical. That's my understanding of it. It's not something that one can have, we have proven, but probably that's what we think is happening.
Thank you. We have a couple more questions. So Sundaram Acharya, who is also part of our team of Biologically Speaking, he's asking, he has a couple of questions. So first question is, the rescue with RAL2 was far from complete. Any hints on other putative regulator from your RNA-seq data? And secondly, he's curious to know the mechanistics of IP3R dominant negative mutants being R or K to Q substitutions.
Okay, I'll answer the first question first, which is about other things. So you are absolutely right. RAL is only one out of the many genes that is downregulated. And in fact, it does only rescue partially. So telling us that there must be other genes that are involved in this whole, that are downregulated and which must be involved. In fact, one of the genes that we had shown earlier, which is, it seems to be important, is the gene that actually synthesizes dopamine. So tyrosine hydroxylase. If you overexpress tyrosine hydroxylase in IP3 receptor mutants, you can also rescue back flight. That was done not in IP3 receptor, but in ORAI, in a sort of ORAI knockdown condition. And you can also rescue. So my suspicion is that there are several genes that are involved in this. And we have tested these two, tyrosine hydroxylase and RAL, but there could be many others as well. And there probably are. We just, we haven't looked at everything. RAL we found interesting because it's a small GTPase. We thought it might be involved in synaptic vesicle release, and it turned out that was the case. So, we could put a function to it. There are others for which we couldn't assign a function easily. Having given such a long answer, now I've forgotten what the second question was. Sorry.
Yeah, he was curious to know the mechanistics of IP3R dominant negative mutants being R or K to Q substitutions.
So what happens is that if you get the substitution, so this ligand binding domain, it has to be in a particular conformation. So this is not my work, okay? This was work done by the mammalian people. And so I'll just try and explain it as best as possible. If you have, when IP3 binds, there's a particular conformational change that takes place in the ligand binding domain, which then allows for the calcium channel, which is on the C terminal, to open. Now, that particular kind of conformation, if you interfere with these three glutamines, it just doesn't happen. Because IP3 binding itself doesn't happen. That pocket into which IP3 binds doesn't form properly. And so there is actually an X-ray crystallographic structure of the N-terminal domain, which has helped people to understand this. Off the top of my head, I don't remember the details, but it's there. And that is what allowed us to actually go ahead and try and make this version. I hope this helped. Yeah.
Thank you. Madhulika Rai, sorry, I'm very sorry. Madhulika Rai is asking, are any of the mutant phenotypes sex specific or age specific? Also, abnormal calcium signaling observed more in males or females?
No. We never found anything that was sex specific so far at all, except that the females are unable to lay eggs. Okay, so that part is sex specific, but that's not so bad. And actually, to be honest, I'm not sure we've really, or at least I don't remember whether the males are fertile or not. But apart from that, none of the motor behavior that I'm talking about, none of that is at all sex specific. In terms of age, it's interesting because flight itself is age dependent. So older flies actually don't fly very well at all. And in fact, that's something that we're now looking at to try and understand what happens to these dopaminergic neurons with age. Do they actually stop functioning properly? What's going on? In a sense, sort of trying to understand this in the context of Parkinson's disease. And this is something that is actually ongoing in the lab. And, maybe in a couple of years, we'll actually have some good answers. So, yeah, I mean, flight itself is very age dependent.
All right. Deepak Korea has a question. He's asking, calcium is widely used as secondary signaling molecule in almost every other cell. He's curious to know if there are some other important functions of the intercellular IP3R in non-neural tissues, for example, skeletal muscle.
Yes, of course. It is definitely required in other tissues as well. It's just that our interest was in, looking at neurons, and flight is almost completely a neuronal phenotype in the context of IP3 receptor. But people have used some of our mutants, in fact, to look at other tissues. And there are other phenotypes for sure. Yeah, I mean, there are epithelial phenotypes. There are phenotypes in fact, right now I'm collaborating with a lab in France, which is, we are looking at it in the context of the lymphatic system and blood cell development. And so, it's something that needs to be studied more carefully, but there are definitely other phenotypes. I mean, the IP3 receptor, I should really say this, and upfront, is it's expressed in almost all cells, and intracellular calcium signaling is probably involved in many things. And that in a sense, that was the challenge, to try and understand it in the context of a specific phenotype, because finally, in the context of human disease, it's the IP3 receptor 1 which seems to be important in the context of spinocerebellar ataxia, and that is a Purkinje neuron specific phenotype. And so, one is interested in understanding it in a specific context, so that one can then think of ways by which you can address it in that context, right? Even though it's probably important in many other things.
Yeah. All right. So last couple of questions. I know we have many more, but just for the sake of time, I'm just going to take 2 more questions. So one from Shweta Bhagavat. She's asking, have you checked for other modes of intracellular calcium signaling other than IP3 in Drosophila, like other species?
I'm not sure what she means by this. I mean, the only other gene that is involved in intracellular calcium signaling is the ryanodine receptor. And no, we have not looked at ryanodine receptor, but the ryanodine receptor does not respond to neurotransmitters and sort of regular signaling processes that are involved in the nervous system. It's more sort of involved in muscle sarcoplasmic reticulum intracellular calcium release. So that's where it comes into play. It may have a role in brain, in the neurons as well, but there are no mutants available, and so no, we haven't looked at that. Yeah.
All right. So the last question for the day is from Shanchari Gorai. She is asking, this IP3 complete blocking leads to larval mortality. Can we use it as means for vector control for malaria or dengue, etc.?
I think it would be tough, because I think what I showed you was that the IP3 receptor in Drosophila and other insects as well is actually pretty similar to what we have it's like 60% homologous to what we have in mammals. And so anything that targets the IP3 receptor could very well also affect humans. So I think this would be a difficult thing to do. I think in general, those kind of targets people do with the genes that are sort of insect specific, because then you are safer in what you're doing. So,
yes.
So probably that may not be the best way to create a mutation and create a sort of insecticide.
I agree. Thank you, Dr. Hassan. With that, this concludes the session. Now I'm gonna hand over it to Shreya for a small vote of thanks. Shreya, if you'd like.
Thank you, Shorodip. Can you hear me?
Yes,
Yes, perfect. Okay, so as we're closing in on the end of these 2 amazing days, I would now like to invite Professor Nahid Ali and Professor Gayati Hassan, our 2 keynote speakers, to close this fantastic meeting that we've had with some words about their experience on this platform, biologically speaking, and also their experience as women in academia. You are our role models, and watching you makes us believe that we too can. So if you can share your thoughts on this as well. I also invite Professor Nahid Ali, who's here with us, to switch on her video maybe and join us.
Hello.
Hello, ma'am.
Can you hear me?
Yes, absolutely.
Hi. So can I take the lead?
I think you should, yes, because I mean, I've been talking nonstop for the last 1 hour, so I think.
So that gives you a break.
Yes, please.
Anyway, I won't take much time. So since we are at the end of these 2 days, and I must say you all have all enjoyed speaking and interacting with the students. and even with the seniors who've been able to join. So what I feel that this is a wonderful platform. And like Geti mentioned, we have not, at least I also can't remember that participating in a platform which is run by our younger researchers and carrying out this conference with a specific theme in their mind. And very interesting, these 2 days have been for women in research. That has been the focus. And it's really nice to give that importance to the women and our young ladies who are working in different areas of research to come up and speak. And it's a lot of encouragement and motivation for everyone, I guess. I think there have been wonderful talks, although I have not been participating in all the talks of the young people across the world. And this platform, especially through Zoom, you are able to connect with everybody around the world. And the time that you chose was probably suitable for India as well as America and Europe. So people could join and interact. And the best part I would like to point out is the interactive session. We normally hear keynote addresses, but that's it. So this gives an opportunity, and by sending their questions, that shyness, hesitation of the young people is also broken. And they can throw their questions to the speakers. I think it's a very good idea, and I really enjoyed it and loved this experience. Thank you very much, the whole group of biospeaking, right?
Professor Ali.
Yeah.
Thank you.
Congratulations, and keep doing the good work.
Thank you. We
Thank
hope to
you.
have you
Thank you.
back for our
Thank
future
you.
endeavors.
I enjoyed it.
Over to you, Professor Hassan.
Yeah, so I can only completely echo what Professor Ali has been saying. It's absolutely, it's a wonderful opportunity to be able to talk to all of you, to be able to talk about our work and to be able to present it. And I think it's wonderful to have, such a interactive question answer session. As Nahid said, there is, when you give a talk in an auditorium, the younger people are always feeling a little hesitant and they don't ask as many questions. And, they meet you afterwards and some of them get to ask. But this seems to be a wonderful way to actually interact directly with them. And I'm really very happy that you've done this, and I'm really enjoyed the discussions. Regarding, continuing in science and doing science, what I would say to all young women and men is that, ask a good question, ask a good question and be passionate about your science. And that's what matters, finally. The rest of it, all moves along. There are always, problems of various kinds that come up. But so long as, you have a passion for the science that you're doing, that's really what matters. And, it's true that women do have a slightly harder time because of various, I would call them actually mostly, situations because of society. At least in India, it's a lot of it is to do with society. There are certain expectations and that makes it difficult. But I think times are changing. And I think one of the most important things is that institutions are now beginning to realize that they need to provide ways by which younger women can actually have families and have careers. And as this change happens with time, it will become easier. And I think, I hope all younger women will, sort of make an effort to be open to the idea and, talking to institutions about how to, help them with managing both family and scientific careers, because that is the big challenge. And I think that conversation is now starting in India quite a bit, in fact, and I'm very hopeful that things will get better with time. So with that, I think maybe perhaps I could, close and say that I hope that was good enough. Yeah.
Yes, thank you so much, professors, for those kind and encouraging words. I just want to invite everyone else on the platform to join in so we can take a conference photo.
Conference photo. Okay.
Yes,
A Zoom
a
photo.
virtual conference photo. Yes.
Sure.
I think they're all waiting in the background.
Right. Yeah. No, it's nice. It's nice to see all of you. Yeah. Good. Yes.
Is everyone here, or are we waiting for a few more people? Yeah.
There's one person whose face is not shown. Ah, there. Yeah. So it's sort of going back and forth.
I think we need to stop the spotlight and just use the gallery.
Exactly.
Yeah. Sorry, we just need 1 more minute.
It's fine.
Yes, perfect. Fantastic. Yeah, thank you so much, professors. We're at the end of this 2-day amazing journey at HerStory of Science. It's been an absolute pleasure to host such a global audience that has stuck with us despite time zones and unstable internet connections. We thank you deeply. Our heartfelt gratitude also to every speaker who's spoken on this platform. They've taken time out of their weekends, vacations, and quarantines in some cases to join us. It's been an honor to host some of our science icons over the last 2 days. Thank you all. We would also like to thank BiasWatch India for choosing us and our conference to sponsor despite some initial setbacks. Thank you, Dr. Vaishnavi Anantanarayan and Dr. Shruti Murlidhar for believing in us and allowing this to happen. Last but not the least, I'd like to thank all members of Biologically Speaking for putting up this show and working behind the scenes with utmost dedication. A huge shout out to our outreach ambassadors, Vani Pandey and Debayan Bonik, for tirelessly working behind the scenes. And of course, the core cast of Biologically Speaking, who have spent sleepless nights to bring this conference to you. Dr. Shaunak Shaho from NIH US, Shoradrit Dash from Vanderbilt University, also in the US, Sanjana Banerjee from the University of Louisiana at Lafayette, also in the US, Dushyant Srivastava from CSIR IICB, Sundaram Acharya from CSIR IGIB, Roopkatha Bhattacharya from CSIR IICB, Krishnamurti Srinivasan, also from CSIR IICB, and me, Shriya Pal Chaudhary from EPFL Switzerland. Thank you all so much. Please keep following us on Twitter and YouTube. Subscribe to our channel on YouTube and visit our website, speakingbiology.wordpress.com, to keep a tab on our future endeavors. We hope to bring you as exciting, as fun adventures as this from time to time. That's all from us today. Thank you all. Have a good morning, good evening, or good night, wherever in the world you are that you're joining us from.
Okay.
And goodbye.
Okay. Bye-bye. Yeah.
Bye.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Thank you.
Thank you so much.
we can open this discussion if anyone wants to talk with us, right? I mean, now it's informal.
Sure.
Yes. Please raise your hand on chat if anybody wants to join our discussion from the participants.
Want to comment anything, give your feedback?
Please feel free to join and share
Okay,
your
that's
feedback.
what I was expecting. I was going to ask if anybody joined, or that was just usual Dushyanta being Dushyanta.
Yeah, we have to uncheck the mute and video option. I guess that will help.
Okay, yeah, that we'll have to do.
Can you stop the streaming? Whoever was